and welcome back. This is our third and final video in our video series for Chapter 3, Cell Division. I'm Miss Delia, and if you're ready, let's get started. So we're having one more look at the slide title and a final look at the learning outcomes. Today we're going to go into 3.3, meiosis, and I hope that you'll be able to achieve these by the end of the lesson. So once again, a recap, we have done 3.1, 3.2, and now we are going into 3.3, meiosis. So meiosis, as you probably already guessed it, it's the cell division process that leads to formation of gametes. So you start out with a cell that has a diploid number, or in humans, that would mean that they have 46 chromosomes. And through the whole meiosis process, you would either get lots of sperm with a number of chromosomes of 23, or you may get an egg, an ovum, and that is also with a chromosome number of 23. So let's look at the stages in meiosis that leads to the formation of gametes. But before that, we need to look at some terminologies. So you need to be able to define chromatid, homologous chromosome, synapsis, bivalent, tetrad, crossing over, and centromere. And that's what we're going to do. So first things first, chromatid and sister chromatid. I've mentioned this before, but let's go through it again. A chromatid is one of two identical halves of a duplicated chromosome. Two copies of a duplicated chromosome attached to each other by proteins at the centromere and sometimes along the arms is called sister chromatid. Dalam arti kata lain, apa maksud definisi ini? What it's saying here? Chromatid is half of duplicated chromosome. Satu chromatid adalah separuh daripada kromosom yang berbentuk X atau berbentuk butterfly ini. Dua salinan duplicated chromosome yang bersambung pada centromere dan kadang-kadang bersambung di bahagian lengan juga adalah digelar sebagai sister chromatid. Dua sister chromatid membentuk satu kromosom. Chromatid akan terpisah semasa mitosis atau meiosis 2. Okay, that's what is meant in the third sentence. While joined, two sister chromatids make up one chromosome. Chromatids are eventually separated during mitosis or meiosis 2. Okay, moving on to the next term. The next term you should know is homologous chromosome. What does it mean by homologous chromosome? It is a pair of chromosomes of the same length, centromere position, and staining pattern. Both chromosomes of each pair carry genes controlling the same inherited characteristics. Dalam arti kata lain, dia adalah kromosom yang berpasangan. Dia punya genetic information tidak sama, tapi dia membawa gene untuk ciri-ciri yang sama. Contoh yang paling hijau, paling atas ini adalah ciri-ciri untuk rambut, tengah-tengah adalah untuk warna kulit, tengah-tengah ini untuk contohnya berapa cepat kuku kamu memanjang, something like that. So these two chromosomes exist as a pair. Memang ini pasangan dia, partner dia. Dia punya panjang, posisi centromere akan sama. One homologous chromosome is inherited from the organism's father, the other from your mother. These are called homologs or a homologous pair. So homologous chromosome, satu kamu dapat daripada ayah kamu, satu kamu dapat daripada ibu kamu. Bersama-sama macam ni, kita boleh panggil dia homologous pair of chromosome, boleh panggil dia homologous pair, boleh panggil dia homologs. Okay, let's move on to the next terms. Tadi itu 2 terms saja, sekarang kita tengok 4 lagi. Okay, so you should know what is the term synapsis. Synapsis is the pairing and physical connection of one duplicated chromosome to its homolog during prophase 1 of meiosis. What this means is, pada prophase 1, okay, in prophase 1, dia akan berdiri tepi-tepi, berdiri sama-sama, dan dia akan berpegang tangan. Okay? Pair and physical connection. This is called synapsis. Bivalent. Bila kita nampak dia membentuk sinapsis, means dia berdiri bersebelahan, kita boleh gelar satu set ini sebagai bivalent, which is satu pasangan homologous kromosom yang melakukan sinapsis. Okay, or a pair of associated homologous chromosomes held together by a complex after chromosome replication. So dia sudah berpegang tangan, kita panggil dia sebagai bivalent. We can also call it as a tetrad. Kita boleh gelar dia juga sebagai tetrad. Tetrad is homologous chromosome consisting of 4 synapse chromatids that become visible during the... Actually, bivalent dengan tetrad membawa maksud yang sama. Dia merujuk kepada sepasang homologous kromosom yang sedang melakukan sinapsis dan crossing over. Beza dia, kenapa kadang-kadang panggil bivalent, kadang-kadang panggil tetrad? Bivalent merujuk kepada berapa kromosom yang ada. Bi means 2. Bivalent means 2 chromosome together. Tetra means 4. Jadi kalau tetrad, dia bukan mengira berapa biji kromosom, dia mengira berapa biji kromatid. So kalau mengikut tetrad, dia adalah 1, 2, 3, 4. 4 kromatid dalam proses sinapsis is tetrad. But really referring to the same thing. And last but not least, crossing over. Crossing over is reciprocal exchange of genetic material between non-sister chromatids during prophase 1 of meiosis. Ini yang ditunjukkan dalam gambar raja ini, di mana dia punya tangan untuk dua non-sister chromatid ini bersilang, dan ada kemungkinan dia akan dapat bertukar. Okay, yang biru dapat yang merah dapat yang biru. This is crossing over. And this is a type of genetic recombination. Macam kalau kita andaikan kita punya DNA itu adalah card, like playing cards, macam daun teruk. Crossing over macam kita sedang shuffle daun teruk kita itu. Okay, it's like you're shuffling your playing cards. And that can be good. Shuffling of DNA can be good. Okay, one more term is chiasma. Chiasma ataupun chiasma lah, mana tahu kamu mau senang hafalkan, kamu mau cakap chiasma. Chiasma is a microscopically visible region where crossing over has occurred earlier. Chiasma means tempat di mana crossing over berlaku. So in this diagram, dia sudah kasih bulat, tempat di mana crossing over berlaku adalah chiasma di sini. Kadang-kadang soalan tanya, ada berapa chiasma dalam gambar raja ini? Jadi kamu perlu pandai kira lah. Okay. Oh, a few more terms. Another term to go is centromere. Centromere is something I already told you in the previous video, but you can read the full explanation here anyway. This is what I would like to focus more on, which is haploid, meaning a single set of unpaired chromosomes, and diploid, or 2N, which is 2 sets of chromosomes are present. One set derived from the female parent and the other from the male. Some organisms can live with haploid cells all over the body. Some organisms can only live with diploid cells all over the body. So, contoh kita, manusia, kita adalah contoh organism yang kebanyakan sel kita mesti diploid, baru kita hidup dengan sihat. Baru badan kita berfungsi dengan normal. So, for us, the normal number of chromosome is 2N equals to 46 chromosome. Tapi dalam kita punya sperm atau ovum untuk manusia, kita akan ada haploid set of chromosomes, which is N equals to 23 chromosome. If you have any issues with this, feel free to ask me, ask your classmates, or ask in the comments below. Okay, let's talk a little bit more about the concept of meiosis. Meiosis is a type of nuclear division which produces 4 daughter cells, each having half number of chromosomes as the parent cell. Meiosis, nuclear division, 4 daughter cell, each of daughter cell have half number of chromosome. And this will occur in gonad cells or reproductive cells of animal which produces sperm and ovum in a process called gametogenesis. There are several phases in meiosis, and the phases or stages are prophase 1, metaphase 1, anaphase 1, telophase 1. And prophase 2, metaphase 2, anaphase 2, telophase 2. Dia masih ikut urutan pro, meta, ana, telo, cuma dia ada tambah satu ataupun dua di belakang. Okay, so these are the stages of meiosis 1 and 2. Starts with interphase, goes until anaphase 1, from telophase 1 into 2 copies. Okay. So, I think if you're having a bit of time trying to catch up, you can pause the video here, have a look at this diagram before we move on. If you're ready, press play, and we're going straight into this one. Kita start terus dengan stage pertama dalam meiosis. Okay, so first stage in meiosis is prophase 1. It is the longest phase in meiosis. This can be further subdivided into 5 stages. Okay, dia sangat panjang. Ada buku yang akan pisahkan kepada 5 stage. Tapi rasanya untuk syllabus kamu tidak perlu hafal 5 stage ini. At this stage, just like in mitosis, the chromatin will condense or thicken and shorten to form chromosome. Sama macam prophase, chromatin DNA kita yang halus itu tergumpal menjadi padat dan dia akan membentuk chromosome. At this stage, synapses will occur, or homologous chromosomes pairing up to form bivalent or tetrad. Dia mula berpasangan dan akan ada crossing over. There will be crossing over between non-sister chromatids which occur at chiasmata. Apa tuh chiasmata? Chiasma, satu crossing over. Chiasmata, 2, 3, 4, 5, lebih dari satu crossing over. Nuclear envelope disintegrate. So, nuclear envelope akan terburai. And centrosome or centrioles move to opposite poles, radiating spindle fibers or synthesizing, growing spindle fibers. Later in prophase 1, microtubules from one pole or the other attach the kinetochores, one at the centromere of each homolog. Apa maksud ayat terakhir ini? Pada penghujung prophase 1, microtubule dari setiap kutub microtubule dari setiap kutub akan bersambung dengan kinetochore. Satu microtubule akan sambung kepada satu centromere pada setiap homolog. Okay, so let's try drawing the prophase 1. I'm not going to do a lot of labeling for this one because I think you already know what things are. So we just draw the cell and make sure you're drawing the chromosome correctly. Okay, so we draw the cell. It's not very nice, but it's all right. I draw the nuclear envelope, which is disintegrating. Nuclear envelope sedang terbuka. I want to tell you that I am using 2n equals to 4. Jadi untuk lukisan kita ini, saya akan andaikan ini adalah sel yang diploid number adalah 4 kromosom. Okay, so I am going to draw 4 chromosome. I start with 1. And I make it pair up with its homolog chromosome. Okay, dia dengan dia punya homolog, dia orang sedang buat synapsis. Sekarang kita boleh gelar pasangan ini sebagai bivalent ataupun kita boleh gelar dia sebagai tetrad. Okay. And then I have another chromosome of different shape. Another chromosome that is the homologous pair. Ini pun sedang buat synapsis dan crossing over. Okay, and then I draw the centrosomes and the spindle fibers. And I also choose to color mine. Okay, saya kasih warna supaya kita nampak di mana ada crossing over. Okay, so that's it for prophase 1. We're moving on to metaphase 1. Metaphase 1 lebih kurang sama, cuma sekarang kamu pastikan kamu menggunakan term homologous chromosomes ataupun bivalent ataupun tetrad. Jangan cakap kromosom saja. Mesti merujuk kepada homologous chromosomes, bivalent ataupun tetrad. They will be arranged on the metaphase plate. Chromosome attached to spindle fibers at the centromere. Spindle fiber sambung kepada centromere. Microtubules attached to their respective centromeres. Ini lebih kurang samalah dua ayat ni. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. The chromatids of the other homolog are attached to microtubules from the other pole. Ini cuma bermaksud kalau ada satu pasangan merah dengan biru ni, merah hanya akan bersambung dengan spindle fiber dari atas. Biru hanya akan bersambung dengan spindle fiber daripada bawah. That's all. Okay, so let's try to draw it now. We draw the cell again. It's okay if not very nice. We draw the centrosomes. We draw the homologous chromosomes lined up on the metaphase plate. So, sekarang saya lukis satu dulu kromosom. Saya lukis kromosom kedua. Okay, pasangan kedua pula. Satu kromosom, dua kromosom. Masih lagi synapsis. So, they're still paired with each other. Crossing over is still happening. And then I'm going to attach the microtubules. Okay, nampak ini di tengah-tengah itu tiada microtubule ya. And then I want to draw the metaphase plate. And I color in the chromosomes. That's it for metaphase 1. Let us go into anaphase 1. Agak cepat kan? Okay. Anaphase 1. This is the stage where spindle fibers will shorten and pull homologous chromosomes, centromeres first, towards opposite pole. Di fasa ini, spindle fiber mengecut, memendek, dan dia akan tarik homologous chromosome. Centromeres first dia means dia menarik daripada centromere menghadap kutub bertentangan. Might be M. Okay, so this will separate the homologous chromosome into two haploid sets. Okay, so sekarang dia akan separate. Nanti disebabkan dia kasih pisah macam ni, nanti kita punya daughter cell akan ada haploid sudah. Itu maksud ayat kedua. Okay, ayat ketiga. Each of the chromosomes still consists of two sister chromatids connected at the centromere. So, setiap kromosom masih ada dia punya sister chromatid. The homologs move toward opposite poles guided by the spindle apparatus. Sister chromatid cohesion persists at the centromere, causing chromatids to move as a unit towards the same pole. Apa maksud ayat nombor 5? Ayat nombor 5 bermaksud sister chromatid masih melekat atau masih berhubung dengan kembar dia. Okay? Still persist at the centromere means sister chromatid tidak terpisah lah. Dia masih berbentuk X atau masih berbentuk butterfly. Okay, let's try to draw anaphase 1. Drawing the centrosome, and we're gonna draw one chromosome being pulled to the left side of the cell. Tengok cara kita tarik ya. Jangan salah arah. Mesti centromere lebih dekat kepada centrosome. Ini pun macam ni, opposite direction. Here's the other chromosome and its homolog. And then I draw the spindle fiber. Sekarang saya kasih warna. Kamu boleh nampak bila saya kasih warna, ada crossing over yang berjaya. Just now they were doing the crossing over, they were exchanging or recombining genetic information. Kamu tengok sini, dia orang berjaya membuat pertukaran maklumat genetik. They are successful at exchanging genetic information. And we will talk about this at the end of this video. Okay. So, itu anaphase, dia akan kena tarik. Dan apa terjadi bila dia sampai kepada kutub pertentangan? Arrive at opposite poles. Let's look at telophase 1. So the chromosomes arrive at opposite poles, and then the chromosomes uncoil and lengthen before becoming invisible again. So dia akan terburai, dia akan jadi kromatin yang panjang dan halus dan sukar dilihat di bawah mikroskop. The spindle fiber disappear. Nuclear envelope will reform around each nucleus, which now possess half the number of chromosomes of the original parent cell. Ayat ketiga, nuclear envelope terbentuk semula. Sekarang setiap ini adalah 23, it's haploid. Okay, so it is, in this diagram, it is 3 chromosome. The arrival of homologous chromosomes at opposite poles marks the ends of meiosis 1. Bila dia sampai di kutub bertentangan, ini bermakna meiosis 1 sudah berakhir. Halving the number of chromosomes has occurred, but the chromosomes are still composed of 2 chromatids. Sekarang dia haploid, tapi dia masih ada 2 chromatid. The nuclear envelope will reform and the nucleoli reappear. Cytokinesis or cleavage furrow in animals or cell plate formation occurs in meiosis 1. So, okay, mari kita cuba lukis dulu. We're gonna draw the cell. We're gonna draw the centrosome, but no more spindle fiber. We're drawing 1 chromosome, 2 chromosome, 3 and 4 chromosomes. I redraw the nuclear envelope, which is reforming, and then I draw the contractile ring. I color in the chromosomes, and I should actually label cleavage furrow, but after this, we are going into cytokinesis. Jadi untuk meiosis 1, ini yang ada beza dengan mitosis kan? Kita nampak sudah dia mula dengan 2N, dia berakhir dengan N number of chromosome. Prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Mesti ada cytokinesis ya di selepas telophase 1. Okay. Kadang-kadang sel itu akan terus masuk kepada meiosis 2. Kadang-kadang dia akan berehat dulu, like after some time baru dia akan sambung dengan meiosis 2. So you have to look at the cell type and the organism, the species, apa semua tu. Okay, so we're moving on. We're going into the next stage, which is prophase 2. Meiosis 2 is a lot like mitosis kan? So dia akan pisahkan sister chromatid saja. In this one, the nucleoli and nuclear envelopes will disappear, and the chromosomes, each composed of 2 sister chromatids, condenses and therefore appear shortened and thickened. Konsep yang sama, daripada chromatid yang panjang halus, dia tergumpal, dia jadi tebal, dia jadi padat, kita gelar dia sebagai kromosom. If there are any centrioles, they will move to opposite poles of the cell, and new spindle apparatus will appear. So now we are going to try drawing that prophase 2. Now your cell will have N equals to 2 number of chromosome. So we draw the cell like this. We draw the nuclear envelope disintegrating. We draw 1 chromosome, 2 chromosome. We draw the centrosome and the spindle fibers. We color in, and yeah, that's it. Ini sajalah untuk prophase 2. Okay, kita teruskan dengan metaphase 2. So what happens in metaphase 2? Chromosomes line up separately at the metaphase plate. The sister chromatids of each chromosomes are genetically not identical. Apa yang maksud poin kedua ini? Sister chromatids of each chromosome are genetically not identical. Kamu tengok dia punya kiri dengan kanan tidak akan membawa maklumat genetik yang sama. It's not going to carry the same genetic information because if you remember in prophase 1 and metaphase 1 we already had the crossing over. Okay, dia sudah bertukar maklumat genetik dengan homologous chromosome. And the kinetochore of sister chromatids are attached to microtubules extending from opposite poles. Itu sajalah. Yang ayat ketiga tu cuma mau cakap dia bersambung dengan microtubule. atau spindle fiber. We're gonna draw the cell. We're gonna draw the cell. We're gonna draw the centrosomes. Centrosome. And we're gonna draw 1 chromosome. 2 chromosomes. And then we attach the spindle fiber. And then we color. Begitu saja untuk metaphase 2. Oh, okay. I wanted to label the metaphase plate. Okay. And then we move on into anaphase 2. Anaphase 2, you will have breakdown of protein that is holding the sister chromatids together, and this causes the centromeres to divide. The spindle fibers will pull the sister chromatids apart to opposite poles as 2 individual chromosomes. Ayat kedua ini yang penting ya. Spindle fiber mengecut atau memendek dan dia akan tarik 2 sister chromatid ke kutub yang bertentangan. Dan bila dia terpisah macam ini, dia dikira sebagai 2 kromosom yang berbeza. Okay, so now this is 1 chromosome, 2 chromosome, 3 chromosome, 4 chromosome, 5 chromosome, 6 chromosome. The centromere will move first, just like in all anaphase. Let's try to draw. Okay, so if we're trying to draw, we're gonna start with the cell, the plasma membrane, and then we draw the centrosome. Kita lukis centrosome. Okay, kita lukis half of the chromosome ataupun 1 sister chromatid, 2 sister chromatid now separated and forming its own daughter chromosomes. Okay, and another 1 sister chromatid, 2 sister chromatid. Sambungkan spindle fiber. And then I choose to color my chromosomes. So this is what it looks like at anaphase 2. All right, final phase of meiosis. We are going into the last one, which is telophase 2. So here the chromosomes uncoil and lengthen, and therefore they become invisible again. The spindle fibers disappear, nuclear envelope reform around each nucleus, and now possess half the number of chromosomes as the original parent cell. Okay, sekarang dia memang officially haploid sudah. The chromosomes are in unduplicated form, and the cytokinesis will occur where it is formation of cleavage furrow in animal cells or formation of cell plate in plant cells. This after telophase 2, it produces 4 daughter cells that are genetically distinct from one another and the parent cell. So the 4 daughter cells are not genetically identical to parent cell. Okay, we're gonna draw this again. We're gonna draw the telophase. Gonna draw the centrosome. Another centrosome. Drawing 2 daughter chromosomes. Another 2 daughter chromosomes on the other side. Drawing the nuclear envelope. Second nuclear envelope. I draw the contractile ring. And I color in the chromosome. And I would like to label the cleavage furrow. After this, after telophase 2, we will have cytokinesis. Okay, so itu sajalah untuk cakap pasal meiosis. Dia terbagi kepada 8 stage. Meiosis 1, 4 stage. Meiosis 2, 4 stage. Kita cakap pasal kepentingan meiosis lah. We talk about what is the significance or importance of meiosis to living things. Well, first of all, it helps organisms that produce sexually to produce gametes by halving the chromosome number. Okay, this is the main importance. And second, meiosis will lead to variation. Variation in nature is good. Okay, why you look different or a little bit different from your parents is because you had variation. Variation can be good because that means, you know what, that's a topic for later. Okay, let's talk about where variation comes from. Okay, the variation is because when the haploid cells fuse at fertilization, there is a recombination of parental genes. Kalau kita guna analogy, shuffle daun teruk, macam saya cakap tu, shuffling playing cards. It's kind of like that concept, but now you're shuffling it with a different set of cards from a different person. Macam kita shuffle down teruk, tapi bukan saja kamu gunakan set kamu sendiri, kamu guna juga card orang lain. Okay, and a second reason why meiosis leads to variation is because during metaphase 1, your homologous chromosomes are together at the equator of the spindle, but they separate into daughter cells independently of each other during anaphase 1. Dia tidak semestinya yang biru semua akan baris sekali. Kadang-kadang biru ada yang sebelah kiri, ada yang sebelah kanan, ada yang kiri, ada yang kanan. So, pada penghujung meiosis 1, dua daughter cell yang kamu dapat sudah berbeza dari segi gabungan genetic information dia. So, this is leading to that. And the third reason why your meiosis has variation is because of chiasmata and crossing over. This is shuffling, separating, and rearranging genes located on the same chromosome. Okay. So, ini 4 adalah kepentingan meiosis. Okay, so here in these notes, you have comparison between mitosis and meiosis. Soalan suka minta kamu bandingkan antara mitosis dengan meiosis. Contoh ini adalah dia terjadi dalam sel apa? Which kind of cell does it occur? Is there pairing or synapsis or not? Does it form bivalent and tetrad or not? Does crossing over occur or not? Apa yang align at metaphase plate? Adakah sister chromatids atau kromosom atau homologous kromosom yang tersusun di metaphase plate? Okay, apa yang terpisah semasa anaphase? Is it sister chromatid yang terpisah ataupun homologous kromosom yang terpisah ataupun macam mana? Okay, cytokinesis berapa kali? Mitosis dia sekali lah. Kalau meiosis, cytokinesis akan berlaku 2 kali. Berapa daughter cell dihasilkan? Adakah daughter cell sama dengan parent cell? Okay, those are all differences that you can talk about. And sometimes this is an essay question. That means you can get a lot of marks if you know how to compare. Kalau kamu tidak hafal diagram, kamu tidak hafal table ini pun, kamu cuba lukis itu mitosis dengan meiosis pun kamu boleh dapatkan perbandingan. Ada juga persamaan lah, bukan perbezaan saja. Ada persamaan, dua-dua pun melibatkan DNA replication. Dua-dua pun perlu pass genetic information to the next generation. Perlu mewariskan zuriat. And third, mitosis and meiosis occurs only in eukaryotic cells. Kalau prokaryotic cells, lain lagi proses dia. Dia bukan digelar sebagai mitosis dan meiosis. Okay, so this is another short summary. Here you can see a short summary about mitosis and a short summary about meiosis. You can read that on your own. And this is my pretty colorful quick summary. Okay, so in this video, we already finished 3.3. We went through some terminologies. We talked about the stages of meiosis. Same thing you can see in this image down here. We talked about the importance of meiosis or significance of meiosis. And we also compare a little bit between mitosis and meiosis. Harap video ini tidak terlampau panjang. Okay, what should you do now? Now, I would like you to take your paper or your notebook or whatever, and please draw the stages of meiosis. You can choose if you want animal cell or plant cell. Dua-dua pun boleh, tapi pilih salah satu lah sebab saya tidak perlu kamu lukis banyak kali. Pilih salah satu, animal cell atau plant cell, lukis diagram dia daripada prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, sepatutnya ada 6 diagram. Lepas siap lukis, label yang perlu saja, tidak perlu label semua. Lepas tu submit dalam GC. And also, go back to the tutorial questions we had for chapter 3. Complete parts A and B in your tutorial exercise for chapter 3, yaitu siapkan soalan tutorial objektif dengan subjektif. Alright, okay. I think that is pretty much it from me. If you want, you can go back. Kaya nanti kamu ada masa, kamu balik kepada slide yang ada gambar blank untuk cell ini. Coba kamu tengok dan coba kamu identify, adakah ini proface kah, interface, ini metaface, ini apa, itu apa. Okay, so go back to your images on that. If you feel like you want more information, feel free to ask me or ask in the group when we have our discussions, or explore resources online. Tiada salah kamu cari ilmu sendiri di YouTube video kah, di website lain kah. It's completely up to you. Alright, so that is the end of chapter 3. We will discuss more in our tutorials, but for now, that's it for me. Thank you very much for your time and attention, and I will see you again soon. Bye!